Good morning. Welcome back to Biochemistry 601. This is lecture number 9, and today we're going to focus on the technique of X-ray crystallography. Now, in the past years, we've actually given 2 lectures about it, but we thought about it and realized most of you are never going to do crystallography, but you are probably going to use coordinates from the protein data bank at some point during your research career. Or if you go to medical school, you're going to understand about drugs and structure-based drug design. So what we're going to tell you today about is how to actually look at a structure and decide whether it's any good or not. Now, most of the depositions in the protein data bank come from X-ray crystallography. And there are some wonderful structures that are in the protein data bank, and there are some that are basic garbage. And that's what you need to understand. Just because it shows up in the protein data bank, and just because the structure has been published in Science, a high-profile, high-impact journal, does not necessarily mean that it's correct. And I'll tell you more about that when we get to what can go wrong in a structure analysis. So today, we're going to turn our attention to X-ray crystallography, but some of the things we're going to talk about how you can say something about a hydrogen bond, or you can't, depending on resolution, is going to also apply to cryo-EM, which is up and coming. That's what everybody's going to do. But the same issues apply to those structures too. They cannot be overinterpreted. So the goal is that you understand the literature and think critically about the structures. There was a professor in the chemistry department who's no longer here. He left to another school. But he was a computational person, and one day he was telling me about all these structures in the protein data bank. Now he's comparing them, and I said, well, have you looked at the resolution that they're solved at? Do you know what the R factor is? And he looked like a deer with headlights coming at him because he didn't realize that there are a lot of bogus structures in the protein data bank. So we want you to think critically. So we're going to talk about the cloning overexpression and protein purification. And that's actually one of the most important parts, because if you get purified protein, chances are you're going to get better crystals. Crystallization, very briefly, tell you how we get the crystals. Then we're going to preparation of heavy atom derivatives. Now that's something that's not done anymore. They use selenomethionine-containing protein. But the reason why I bring it up is because when I tell you about that, what can go wrong in an X-ray analysis, they were using heavy atom derivatives. And they basically, when you see that, they're trying to get phase information. That's what they're trying to do. X-ray data collection and processing, that's what you have to go through. Determination of your selenomethionine positions or your heavy atom positions. Calculation of these so-called protein phases. Interpretations of electron density nets. Now that is important, and refinement of the structure. And I can tell you that there are some coordinates in the protein data bank that have never seen human eyes, that they're built automatically by a computer program and they're deposited in the protein data bank. And let me tell you, I look at every structure that has come out of my laboratory, and I can tell you that sometimes what the computer program has built is just nonsense. So, in Canvas, or on Canvas, there are these 4 articles. Of course, you're not expected to read them and memorize them, but they may be important someday in your research. One is called "You Are Lost Without a Map, Navigating the Sea of Protein Structures." Very good article written by a friend of mine, Nicholas Slovaki. There's protein crystallography for non-crystallographers, or how to get the best, but not more, from published macromolecular structures. That one's good too. There's another one, protein crystallography for the aspiring crystallographers, or how to avoid pitfalls and traps in molecular structure determination. And you can see that people are starting to get mad at some of the stuff that's there. Here's avoidable errors in deposited macromolecular structures, an impediment to efficient data mining. So these are worth reading if you actually get interested in this field, if you're interested in protein structure for a career. So that leads us to this structure, myosin subfragment 1. This is the one that Ivan solved. I helped him build the model, but he's the driving force of being able to figure out how to get the crystals and get the data collected. It was an amazing thing. It took maybe 10 years. It was really difficult back then. So these are really pretty pictures, and they show up in textbooks, they show up in slides of people's seminars, but the question is, what can you get out of them? How do you think critically about them? How do you interpret them? Because some are way over interpreted, way over. So you gotta always ask questions. These are pretty pictures, they are helpful, but they're not going to tell you everything that people think that they do. So here's the practical route to an X-ray structure. You've got to identify, express, and purify the macromolecule that you're interested in. Now that's a whole lot easier. You just go get a gene and clone it, and if you're lucky, you get it overexpressed, the protein overexpressed, you purify it. Back when I started, this didn't happen. So, for example, the enzyme I worked on, we had to go get 80 pounds of pig hearts from the slaughterhouse, and you had to dissect the hearts, and then there was all this stuff. It took 3 weeks to get the protein purified, and then you'd crystallize it, and then you'd go back to the slaughterhouse. Well, it's much easier these days. Obviously, I had middle school students doing it. So, in a way, we can pick what we're interested in now, and we can say, I'm interested in that, as opposed to, can I get it out of pig hearts? Then you crystallize the macromolecule, and that doesn't always happen, and that's really sad, but it happens, it happens. You record the X-ray data, you determine the crystal structure, fitting the model, which is my favorite, what I'm doing right now after lecture, interpret the meaning of your structure, and that's important. So let's take a look at the crystallizations. It's achieved by reducing the solubility of the protein without it precipitating out. You want crystals, you want an ordered array of molecules. And so there's no set recipe for it. Each protein differs. And what's interesting is you can make a site-directed mutant in the interior of a protein. And for example, this happened to us with the cytochrome C. There was a tyrosine in the middle of that molecule. We crystallized it, we solved the structure, and then we were interested, is that tyrosine important? So we changed it to phenylalanine in the middle of the protein. Now you would think it would crystallize under the same conditions. It did not. So if you're looking at a protein and somebody says, well, the homologous one's been done that way, and they use those crystallization conditions, don't even bother listening. Every protein, even site-directed mutant proteins, will crystallize differently. And there are cases when we've grown crystals, and the crystals aren't very good, but we were able to get a rough structure, and we could see there was a residue like aspartate that was important for a crystal lattice. So we actually mutated a surface residue and got better crystals. That's something people do a lot of. So you have to figure out what precipitant you're going to use, and there are some precipitants that are much better than others. You want to figure out the optimal technique, and usually now it's hanging drop, but there are other ways to do it. And here's something, crystals contain a lot of water, 50% typically. So people have this idea that protein crystals are packed really tightly. They aren't. They aren't. And in fact, proteins, enzymes, are active in the crystalline lattice. I can't tell you how many times we've soaked in substrates into our crystals, and it turned over and gave us product. And when it isn't active in the crystalline lattice, it's usually because of the way the molecule is situated in the lattice, the substrates can't get in. So there's huge areas of solvent. So there's always what you see in the crystalline lattice. Is it really true in the cell? Well, of course, we're all reductionists. When you open up that cell, you become a reductionist. But I can tell you that enzymes are active in the crystalline lattice, and there's a lot of water. So that's important. Now, you can go commercially and get all these different types of crystallization screens. Example is the Hampton research. So it's much easier than it used to be. You don't have to make up all these solutions. You can go to the screen. The problem is, once you get a hit on those screens, a lot of people don't know how to get the crystals bigger. And the bigger the crystals, the stronger the X-ray data, and the better the structure is actually going to be, under most cases. So these are the important parameters in protein crystallization. The state and purity of the protein. If you want a chance to get really good crystals, your protein has to be pure. It has to be pure. And that's important. That is a lot easier these days with these his tags, all that kind of stuff. And here's another thing. When we look for protein crystals, we will actually put a his tag at the N terminus, and that's cleavable with TEV protease. So we'll have an example where you have a his tag on the protein, or the his tag removed and the protein, or a his tag at the C terminus. So we have 3 different variants of the protein that we set up, and you'd be amazed. Sometimes the tagged protein will crystallize and the tagless won't. Or sometimes the tag at the N terminus you'll get protein crystals, and sometimes at the C terminus you will. So there's all those things you need to think about as well. Precipitant, type and concentration, and I'm going to mention those on the next slide. pH, it's important. Now we usually test it in half pH units because that's about what you're going to see. I'm amazed when I see crystallizations and they'll say it was pH 7.23. Are you kidding me? It's either pH 7 or 7.5. It's basically, you go in half pH units. That makes a difference. And why does it make a difference? Because sometimes there might be a residue at the surface that when it gets protonated, then the protein can go on the crystalline lattice. But as long as it's not protonated, there's a minus charge, like an aspartate or a glutamate, then it can't crystallize. So pH is very important. I've seen when we've set up crystallizations, you'll see all of a sudden some little crystals, maybe at pH 5, bigger crystals at pH 5.5, great crystals at pH 6, and then not so good crystals at 6.5. So pH is really important. It's also important that you read it because, no, I'll get to that. All right, temperature. We like to set it up room temperature because nobody likes to go sit in the cold room. But sometimes you have to go into that cold room and set it up because the protein will only crystallize in the cold room. Protein concentration we'll talk about. This presence of substrates or products or analogs, that's very important in enzymes because what happens a lot of times, like UDP galactose 4 polymerase, it's bilobal. The N-terminal domain is that Rossmann fold that binds NAD. The C-terminal domain binds the substrate. And without substrate in there, there's kind of this floppy motion going on. And once you put the substrate in, or product analog, it basically tightens down, and then you get crystals. So that's important. Do you add DTT or EDTA? Things like that. Time. Modification of surface residues. And as I said, with the myosin subfragment 1, nobody could get crystals until Ivan methylated the lysines at the surface. So we do this kind of stuff now too. We'll do the methylation reactions. Or as we said, sometimes we get a rough structure, maybe at 3 angstrom resolution, and you see, well, if I change that little patch of negative charges up there, will I change the crystallization properties? So we do site-directed mutagenesis. And the source of the protein. For example, if you get it from one bacterial source, like E. coli, and you don't get crystals, or the crystals aren't any good, you change source. For the same enzyme, go to Lactococcus lactis, whatever, change the source. Now these are the common precipitants that you see. Ammonium sulfate, ammonium phosphate. I don't need to read this list to you. These are high salts. These are okay. If you get crystals, that's fine. But one of the things you see when you're looking at enzymes, that a lot of times if the enzyme is crystallized in high salt, when you try to get a substrate bound, or a product bound, or a product analog bound, they will bind, but not in the physiologically relevant position. There's been a lot of things misinterpreted. So high salt, if that's the only way you can get the crystals, so be it. But we tend to want to use these organic precipitants, including polyethylene glycol 6,000 to 8,000. These, if we get crystals, we're jumping for joy if we have it in these organic solvents, organic precipitants. And then there are some alcohols too. So those are the types of precipitants that you will see. High salts, okay, if that's the only way you can get it. But if you can get your protein to crystallize in, for example, polyethylene glycol 8,000, you're going to be better off. Now the effect of temperature. After precipitant, pH, temperature is by far the most significant. pH is important. That's important, and the precipitant. But most crystals have been grown at 4 degrees. More crystals have been grown at 4 degrees Celsius than at room temperature, which means you have to go into the cold room, and sometimes in the middle of winter here with a polar vortex, you don't want to be sitting in that cold room for an hour, but that's what you have to do. Some proteins will only crystallize at 1 temperature. So we've had times where crystals won't come out in the cold room, but they come out at room temperature. So that's another thing to think about. Proteins are typically more stable at lower temperatures, but that's hard to say. That's hard to say. And a stable environment is important for growing crystals. What does that mean? It means when you set up your crystallization trays, you put them away and you don't rattle them. In fact, you have to be careful because people are so excited, they come down, they want to see if they grew crystals. Leave them alone. Leave them alone for a couple of days. Now, after a couple of days, if you see you got some small crystals, then you set up around that region and you leave them go. Let them go for a week or two. Right? But be careful. It's a stable environment. Now, if you ever collaborate with a crystallographer, they're going to ask you how concentrated can you get your protein. About 10 to 20 mg per ml works fine. Now, you can get crystals at 1 mg per ml or 5 mg per ml. It really depends. But what you typically see is 10 to 20 mg per ml. And that's important when you're actually looking with salts, like ammonium sulfate. You'll tend to have the protein concentration higher. In polyethylene glycol 8,000, it typically is lower, like 5 mg per ml, but there's no set in stone here. But that's important. You need to be able to get your protein concentrated enough. Now, one of the proteins I worked on when I was in Oregon was actually thermolyzing. That was 400 megs per ml. So you can get that concentrated. As I said, it's generally the lower for crystallizations from polyethylene glycol 8000. The protein concentration is not the major determinant, but it certainly can help. But what is really going to help is your protein, the purity of your protein, and the pH. Now, as I mentioned, substrate products and analogs with 50%, more than 50% of the enzymes, we find that you get better crystals if you have some ligand bound, as I told you for UDP galactose 4 epimerase. And that's basically what we need to talk about in this slide here. But this is important. If you solve the structure of an enzyme, that's great. You can maybe have an idea about the active site because you've done things like EDC or CMC, that kind of stuff, right? But it's better if you have a product analog or a substrate in there, or some kind of ligand, because that'll help you see where the active site is and will give you a clue of which residues are important in catalysis. Now, additives are important. Sometimes you get these little crystals and you can't get them bigger. Rather, they're just too small, or they're forming these like layers of crystals. So you add additives, and sometimes the additives. So there's a wide range of small organic molecules, like DMSO, for example, that have been used to get bigger crystals, or ethylene glycol, or MPD. And the choice of buffer can be important. So you're really, by now, starting to realize how many variables that you have. And for example, you always purify your protein the same way, or otherwise you're going to have trouble. So there was a technician in the lab where I got my PhD, and he purified the protein, and he set it up using polyethylene glycol 8000 solutions that were buffered. And then one day, he decided, well, I don't need to buffer those. Well, all of a sudden, he started getting a different crystal form. So you have to be careful. The choice of buffer. Here, for example, is a structure we saw where you could see the MOPS buffer in the electron density map. So the choice of buffer sometimes can help bring molecules together to get that crystalline lattice. So, typical crystallization mixtures contain a whole lot of stuff. And that's why you have to be very careful when you take concentrations. Make sure you do everything the same way. Here's another example. I was growing these crystals of this cytochrome. I would crystallize it. I always got these zillion little crystals. I couldn't get those crystals bigger. And then one day, I went to dilute the protein down with the phosphate buffer I had made. But this time I grabbed the wrong bottle. And that phosphate buffer also had sodium chloride in it. And I set it up, and then I looked at it and I realized what I had done. But I left the crystals alone, the crystallization trials alone. And about 2 weeks later, I came back, and these crystals were big enough you could make a ring out of them. So be very careful that you're setting up stuff, you know what you're doing. Now this is important. Always, when you read a crystallography paper, and there are 2 reading assignments at the end of the semester, that you need to take a look at the information about how the crystals were grown. In particular, there's one structure. And had the reviewer seen this, maybe they could have stopped kind of an embarrassment in the science. But you'll see that paper. Right, so this information should be included in the publication, and it should be found in the protein data bank. Now, unfortunately, these days, people aren't interested in details, so a lot of times you have to hunt around for the crystallization conditions. But you need to know, for example, what pH these crystals were grown. Because if you have an enzyme and you grew the crystals at pH 4, and this enzyme is not active at pH 4, and they're trying to talk about a catalytic mechanism, you have to ask what's going on. Now, why would anybody grow crystals at pH 4? Here's an example. A fatty acid binding protein that we crystallized. We could never get those crystals big. And so we kept dropping the pH, dropping the pH. We got to pH 4 and all of a sudden beautiful crystals. So we solved the structure. And after you looked at the structure, you realized that there was one fatty acid binding protein sitting next to another one in the crystalline lattice, and there was an aspartate and 2 aspartates facing each other, and they had to be protonated for the crystalline lattice to grow, to occur. Right, so pH is important, but when you read a paper, make sure you look at the pH, see what's going on. Now, no structure, or a structure, is better than no structure, but you have to be careful. Experimental detail is important. Unfortunately, it's been put into supplementary material. It is not supplementary. It should not be supplementary. There are different types of crystal screens. Most use this sparse matrix screen, where in fact you get these kits from Hampton, and it's screening a wide variety of crystallization space with different pH, different organics, different salts, all types of things like that. And it's good, it's random, but the issue is once you get small crystals, a lot of times people don't know how to translate the information from the Hampton screen back to growing larger crystals. And the larger the crystal, the better your X-ray data is going to be. Now there's this systematic one where you vary one parameter at a time. So maybe you have your protein and you're going to say at pH 7 I'm going to look at it with a 5% PEG, 10% PEG, 15%. It's more systematic. Well, that's the way we used to do it, and to be honest, it works okay. What you really want to do is use the sparse matrix screen and get a hit where you think the crystals are going to appear. And then you use the systematic. You know generally where it's going to be, and then you start systematically varying the buffer, for example, or the pH, or the precipitant concentration. Crystallization, it's time dependent. There's one time when I set up crystals of a dehalogenase, I went to get a cup of coffee. I come back about 45 minutes later and there's crystals growing all over the place. It happens that fast. Sometimes though, they take a long time to grow. Right? On occasion, the crystallization can take months. Carbanophosphate synthetase, the structure we saw, took months. There's always a problem though if something takes months because it means something's happening. Maybe your asparagines are deaminating. Something's going on. Maybe the protein's getting proteolized by a contaminating protease. Something's going on. All right? Maybe there's oxidation going on, or the deamination of asparagines and glutamines. So if there's a problem, you have to think about it. One of the dehalogenase structures that we saw, it was amazing. So it had CoA in it, okay? And CoA, we'll talk about CoA when we talk about cofactors, has a phosphate group hanging off a ribose. We could never get the crystals as big as we would like. But we finally solved the structure, and when we solved it, we saw that the CoA was not the phosphorylated version, but there was D-phospho-CoA, which is a contaminant of the CoA solutions that you buy from Sigma. And so what was amazing is once we bought D-phospho-CoA, you could grow these absolutely huge crystals. So, let's move on. Crystallizations. These are the old-fashioned dialysis and batch. Nobody uses those anymore. Well, I shouldn't say nobody. That was a hyperbole. People still use batch. Some people will still use dialysis. It comes in handy sometimes. But most of the time these days, it's used by vapor diffusion. And if you want to read more about this, Introduction to Protein Crystallization, this is a good article. And in fact, Alex McPherson is the one who set up the Hampton research screens. It's a good article. He's a good crystallographer. So the vapor diffusion method was introduced back in the 1960s. And it involved basically drying down your solution. But this conventional sitting drop method was introduced by Bob Bach, who was our graduate school dean here at the University of Wisconsin. And Bob Bach was the dean when I got my job here. He was a really neat person. He would actually walk down, you'd be walking back to class or something, and he'd see you. He knew your name, he knew what kind of research you were doing. It was really neat, Bob Bach. So the hanging drop method, which is what's used these days, was developed in 1971. So briefly, the hanging drop method of vapor diffusion. You have your precipitant here. You have a cover slip. You take a concentration or a aliquot of your protein, maybe 1 microliter, and you put it on the cover slip, and you take 1 microliter from the precipitant down here and you mix them together so it's a 1-to-1 mixture, and then you flip it over and cover with oil here, seal it with oil, and then over time by vapor diffusion, the water will come down here and the protein will the drop will start to basically dry down, and if you're lucky, you get crystals. Now, the bigger the droplet tends to give you bigger crystals, but people use really like 1 microliter. We used to use 10 microliter. This is probably the most common way, so vapor diffusion. Now, the disadvantages, it's hard to control crystal growth, so sometimes it starts to come down and then you get a zillion of crystals coming out, and really you want 2 or 3 crystals per droplet. But it's easy to perform, you use small volumes, it's easy to nucleate. So that's the method. So when you read a paper, it'll say they were grown by vapor diffusion, hanging drop method of vapor diffusion. Now, You go in and you're looking at your crystallization trials and your trays, and you're all excited, and you got a crystal. Well, you got to be careful, because sometimes you get crystals of small molecule. For example, magnesium phosphate, they make really pretty crystals, but the structure of magnesium phosphate is already solved. So the test, the X-ray diffraction, is the best test for whether you have crystals or not. And here's another thing that's sad. Sometimes you grow these absolutely gorgeous crystals, and you're jumping up with a wall because you've got these beautiful crystals, and you put them in the X-ray beam, and they don't diffract. And then you're sad. So you have to, once you get your crystals, and you're excited, you need to put them in the X-ray beam, and make sure that they actually diffract, and diffract well enough to give you the information you want. So there you go. That's a diffraction pattern. Now let's talk about the nature of crystals. All right, they're precisely ordered arrays of molecules. Precisely ordered arrays of molecules. It's a solid which possesses long-range, three-dimensional order. And the asymmetric unit, that's an important thing that you're going to hear. Asymmetric unit, it's the smallest, unique portion of the crystalline lattice that once you know what's in the asymmetric unit, then by the crystal symmetry, which we'll talk about, which is dictated by the space group, you can generate the unit cell, which we're going to talk about. And then once you've generated the unit cell, you can move the unit cell in 3 dimensions to generate the lattice. Asymmetric unit, important concept. So, the other thing, basic definitions. When you're looking at a paper, you want to take a look at the unit cell dimensions, because typically the longer the unit cell dimensions, the more difficult it is to collect the X-ray data. That's a given. Now, viruses, for example, the unit cell dimensions can be 500 to 1,000 angstroms. The bigger the unit cell, the more volume there is of protein in there, and sometimes the more difficult technically it can be to collect the data. But with synchrotron sources these days, it's much easier than it used to be. So when you're reading a paper, check out the unit cell dimensions. They will also tell you that the crystal belongs to some space group. And how do they know what space group it belongs to? It's the diffraction pattern from the crystal. It's the way the diffraction pattern looks that they can determine the space group. And a space group is just a description of the means by which a fundamental set of asymmetric units are self-related. I'm going to show you a picture of that in a minute. Now, we have calculations, not we. We know from calculations that there are exactly 230 different possible combinations of symmetries that can pack identical copies of objects into a 3-dimensional lattice. 230. And in fact, I forgot to bring down the book. It's in international tables from crystallography. There's the 230 space groups there. They tell you what the symmetry properties are. It's everything you want to know. Right? There's 230 different ways. Now for crystallography, for protein crystals and nucleic acids, we don't have to worry about 230, and I'll talk about that in a second. We only have 65 we have to worry about space groups. But when they say the space group is P21, they're just telling you how one asymmetric unit is related to the other asymmetric unit in the unit cell. So going from the asymmetric unit to the crystalline lattice, this is our asymmetric unit. Assume that's the protein, and we'll talk about what you can have in the asymmetric unit. Supposing you have a monomer, right? Then the unit cell, in this particular case, you solve that structure by the twofold that's dictated by the space group. You can generate the other asymmetric unit. This particular unit cell, and it's a 2-dimensional here, has 2 asymmetric units that are related by a twofold. So once we've traced this polypeptide chain, we can then trace that one by a twofold rotational axis. It just puts it over there. And then you can take the unit cell, and in this example, of course, it's 2-dimensional, but you can translate the unit cell in 2 dimensions to generate the lattice. And that's exactly what you're doing in protein crystallography. Now, what's in an asymmetric unit? And that's something you want to look at when you're looking at a paper. An asymmetric unit, for example, could contain 1 monomer, or it could contain 2, or it could contain 3, or it could contain 4, which means you have 4 in the asymmetric units. They're individual molecules. But if there's 4 in the asymmetric unit, once you've traced all those 4 chains, then by the crystal symmetry, you can generate the rest of the lattice. But what happens if you have a trimer? Supposing you have a trimer. You could have a trimer in the asymmetric unit. You could have 2 trimers in the asymmetric unit. Or you could have one subunit in the asymmetric unit if the trimer, with its threefold axis, basically aligns with a crystallographic threefold. So let's talk about a tetramer. Maybe you have one tetramer in the asymmetric unit. But you could have half a tetramer if the twofold about half the tetramer aligns with the crystallographic twofold. Or you could have one subunit. Insecticide, when we saw that structure, it's a tetramer. But we only had one molecule in the asymmetric unit because the two twos aligned along the crystallographic twofolds. And so we ended up reducing the asymmetric unit to 1. We know that there are 7 crystal systems. These are the 7 crystal systems. Of course you don't have to memorize it. But when you're reading a paper, they may say, well, it belongs to the monoclinic space group P21. Or orthorhombic. Orthorhombic is my favorite because the unit cell dimensions A, B, and C are not equal, and alpha, beta, and gamma are 98. Remember that unit cell is the thing that you can translate around to make that three-dimensional lattice. Hexagonals are neat. So here are the essential symmetry of the crystal, the unit cell dimensions, and cubic lattices are the most symmetric. And then the tetragonal lattices have 4-fold symmetry. Hexagonal lattices have 6-fold. Triclinic lattices have no rotational symmetry. So all it is, it's telling you, well, it belongs to that monoclinic space group. Now, what are these space groups? Well, they're symmetry operations that relate one asymmetric unit to the other asymmetric unit, to another one, depending, maybe you have a crystalline lattice, the unit cell has 4 asymmetric units. Now, what we see in proteins is you can have a 2-fold, a 3-fold, a 4-fold, and a 6-fold rotational axis. You don't have 5-folds. You can also have what's called screw axis. So one asymmetric unit might be related to another asymmetric unit in the unit cell by a rotation and translation. We can't have mirror planes, reflection through a plane. We can't have glide planes, which is reflection and translation. And we can't have centers of symmetry in protein crystals or nucleic acid crystals. And why? Because we have chiral molecules, because we have L amino acids. So that's why, instead of 230 space groups that we have to deal with, if you're an inorganic crystallographer and you're solving structures of inorganic compounds, you have to worry about 230. We don't. We only have to worry about 65. And those 65 are listed on this slide here. Now, what you see when you read a paper is not all these space groups are equally represented in crystal structures. There are a few that tend to be common, like C2221 is the one I love. This one, P21. This one is one that you see a lot. It's a monoclinic here, and we actually named our first cat P21. But we would take it to the vet, and it kept saying it's P21. It's like, no, it's P21. In fact, the vet said, what do you guys do for a living? All right, so anyhow, our first cat and our best cat was named P21. But there's tetragonal and trigonal. So just read the paper, and what it's telling you is how the asymmetric unit is related to another asymmetric unit in the crystalline lattice. Now let's talk about some of the symmetries that you find in biological samples. All right, so this is a 2-fold. We have 2 folds in our crystalline lattice. You're looking down the local 2-fold, or the crystallographic 2-fold that relates that asymmetric unit to that asymmetric unit, or B to B. Here's a 3-fold, so you're relating 120 degrees, 120 degrees, so this is 180. Now these screw axes, what happens is, you can have an asymmetric unit related to that asymmetric unit by a rotation of 180 degrees, and then a translation half the distance along the unit cell. These are the kind of symmetries that you see. Now I'm going to show you some examples. So this is 4-chlorobenzoyl-CoA dehalogenase, and I talked about this before. This particular protein crystallized in the space group P21212. What does that mean? It's a primitive space group. It has these 2-fold screw axes, so it's a rotation and a translation, and this is just a 2-fold. The 2-1 are screw axes. And it means you rotate 180 degrees and you translate it a half along that unit cell edge. And as I told you, this enzyme does this reaction, a nucleophilic aromatic substitution, something the organic chemist would die for. And when the structure was solved, we had a whole trimer in the asymmetric unit. This is the case where you're looking down the local 2-fold, 3-fold rotational axis that relates that subunit to that subunit to that subunit. And you also have domain swapping going on here, and the active sites are shared between 2 subunits in that trimer. Now here is an example. This is methanyl-malonyl-CoA decarboxylase. This particular protein crystallized in the space group C2. And what does it do? Well, as the name implies, it's knocking off a carboxylate group here. This is what we have in the asymmetric unit. It's a trimer. Looks like a trimer to me. Here's one subunit, another subunit, another subunit. But you don't see the same kind of domain swapping that you saw with the dehalogenase. Looking down, that. So this was the asymmetric unit. That's the asymmetric unit. Now, let's take another look at it. This is what we had to trace. So we had to trace each one of those chains. And once we had done that, then by the crystal symmetry dictated by that space group, we could generate the whole unit cell in the crystalline lattice. Now, if I turn this molecule 90 degrees, so this blue now is up here. Well, that's interesting. But now, if you look at way this packed against another molecule in the crystalline lattice, you see that there's a twofold rotational axis. That's the crystallographic twofold. That's it. But there's all these interactions going on, all these interactions. And when we did ultracentrifugation experiments, you find out that this protein migrates as a hexamer. It's a hexamer. So here's an example in the asymmetric unit where you had half of the hexamer because you have a 3-2 symmetry and the molecule packed. So you had a local threefold that I showed you on the previous one that's intersected by this twofold that's crystallographic. So that hopefully explains a little bit about asymmetric units. Now, why do we use X-rays? Why do we use X-rays? And this figure comes out of this wonderful book by Dennis Sherwood, which is out of print. I actually brought it along. It's everything you'd ever want to know about crystallography, and probably more than that. It's a really good book. He does a wonderful job of explaining this stuff. And this is from his book. Now, think about this. You have a boat out in the ocean, and it's 5 meters in length. And then you have the waves. And the wavelength is about 20 to 30 meters. Supposing you're standing on the shore and it's foggy, right? The boat, basically, the waves are unperturbed by the boat because the wavelengths are longer than the boat. So you'd never know there's a boat out there if you were just watching the waves because the waves are unperturbed by that boat. But if somebody dropped a pebble and you start getting diffraction back here and you're standing on it, you would see that. You would see that because now the waves are smaller than the dimension of the boat. And that's why we have to use X-rays. So these are the different wavelengths, right? The size of people here, right? Radio waves, microwaves. But if you get into here, X-rays, all right, that's where we're starting to get down into angstroms, like 1 angstrom. Well, what's the carbon-carbon bond length? 1.54 angstroms. So we have to use X-rays to do this because otherwise the object would be unperturbed. If we took a crystal, right, and we put it in something like a microwave, the microwave would just pass through unperturbed. That's why we have to use X-rays. So what is diffraction? It's the process by which matter deflects light rays from their straight path. It's diffraction. And in order to get that with the crystalline, we have to get down to 1 angstrom resolution, 1 angstrom wavelength, or 1.5. The precise pattern made by the scattered light is called the diffraction pattern. And the diffraction patterns of protein crystals are beautiful. They're beautiful. There's a lot of data there. Now let's just look at a light microscope. And this particular figure is old, and sometime I'm going to have to remake this one. But I think this explains pretty much what's going on. You start off, for example, an amoeba. Now, if you go back to this slide here, if we go back to the slide here, visible light, we can see something like an amoeba. So, if we go to here, we've got this amoeba in our light microscope. We put visible light, it hits the amoeba, and the light rays are diffracted. And then there's this lens that refocuses these scattered light rays onto an enlarged image, which is shown here. And then there's a magnifying lens, an eyepiece lens, that further magnifies it, so you get this enlarged image of the object. So you can do this with visible light if you have something about the size of an amoeba, for example, light microscope. Well, how does the X-ray crystallographer do it? All right, first thing is you have to crystallize your protein. And why do you have to crystallize your protein? Because of signal to noise. You've got to get all those protein molecules lined up so that when the crystal diffracts the X-rays, you can get some information out of it. So you have your X-rays, you've got your crystal. Then you collect the X-ray data as that crystal scatters the X-rays, and it's a three-dimensional thing that goes on. You collect the data, but here's the rub. There's no known material that will refocus X-rays. You can't refocus them. So what are you going to do? Well, that's where the crystallographer steps in. With our computing, we act as the lens. We take these scattered X-rays, we get information about the intensities of these scattered X-rays, and then by a lot of fancy footwork and computer programs and preparation of heavy atom derivatives, selenomethionine, those kind of things, we are able to actually refocus those X-rays by our computation called Fourier synthesis, actually, to give this electron density map. And that electron density map is a roadmap that allows us to look at what the protein looks like in three-dimensional structure. So here's an old-fashioned electron density map. And you can see, well, you can't see it if you were actually in class, you'd be able to see it, but you can see the electron density. It's really neat. It's a roadmap. It tells us what the protein looks like in 3 dimensions. So here's an updated slide. You have your crystal. You hit it with X-rays. You collect lots and lots of data, lots and lots of data. You use the computer and graphics. You get your electron density map, and then you build your model. It's kind of fun. So let's talk about the review of behavior of light. Now, when a crystal is illuminated with X-rays, each electron in the object oscillates and re-radiates the X-rays. Each electron. You got a lot of protein in those crystals, and you got even more electrons. And the diffraction pattern seen by a distant observer is the sum of all the X-rays being scattered or diffracted by the crystal. If you drop 2 pebbles in water at different places, you obtain 2 intersecting ripples. Now, an observer at the surface of the water sees the sum of the 2 waves. If the crest from one wave falls on the crest from the other wave, the resultant wave is twice as high. If the crest falls on the trough, the resultant wave vanishes. And that's what you see with these patterns, these diffraction patterns. There's only specific places where you get constructive interference. So when a crystal is illuminated by X-rays, every electron in the crystal re-radiates a spherical wave analogous to the circular wave caused by the pebbles. So here we go. Just if you look at 2 waves, if they're perfectly in phase, right, you get this doubling. If you add them and they're not in perfect phase, you get that kind of wave. But in the case where there's destructive interference, they're completely out of phase, 180-degree phase, you won't get anything. You won't get anything. So here is an example of a precession photograph that was taken in an X-ray lab, actually our lab. Now people don't use precession photographs anymore, precession cameras, and it's fine. It's fine. But it shows you a slice of the diffraction pattern, and the patterns that you see in each pattern is going to be different depending on what protein you've crystallized. So the diffraction pattern is telling us what that protein looks like in 3 dimensions. Every point on this X-ray film is struck by a wave that is the sum of all the diffracted X-rays emanating from all points within the crystal. And you can see where you have constructive interference and destructive interference here. And it's that pattern, the diffraction pattern, that allows a crystallographer to determine what space group the protein crystallized in. Here is an example of an oscillation photograph where you put the crystal in the X-ray beam and then you oscillate it back and forth by a degree. And then you collect another oscillation photograph as you're moving the crystal along. But this diffraction pattern is coming from all the electrons in that crystalline lattice. And it's that information. The problem is, with that information, we lose all information, unfortunately, with respect to the phase. And in order to calculate an electron density map, you have to have information about the intensity of those X-rays, or the diffraction pattern. But you also have to have phase. And every one of these diffraction maxima has a phase associated with it, and that's the information we lose out on. We can't refocus those X-rays, And because of that, we lose information about the phase. And so we have to get phase information that allows us to actually calculate an electron density map. Now these days, most data is collected at synchrotron sources. We have our own X-ray equipment, but a lot of labs don't. And so you go to these sources, and it's really fun. It's actually fun to do it. There's Argonne National Laboratories, that's the one we go to. There's Stanford and Berkeley, Cornell, Brookhaven, and Baton Rouge. But it's huge facilities. This is the synchrotron source right here, and you actually have bicycles that you can cycle around the different sectors. So you apply for time, and then you can go collect your data. But you're only given a limited amount of time for maybe a day or two, so you basically stay up the whole time. Now usually when we go, my senior scientist is the one who stays up, and I'm the designated driver, so I actually go stay in the guest house and get some sleep. But it's fun. There's something really exciting about collecting your data. Now, what's this stuff about phases, and what's this stuff about the crystallographer being the lens? The lens that we use, right, is the Fourier synthesis. The Fourier synthesis. So in microscopy, the diffracted light waves are added back together by the lens, producing an image. The Fourier series is a mathematical equivalent to that lens. But in order to do that equation, we have to have information not only of the intensity of the scattered X-rays, but their phases. And that's the so-called phase problem. And here's an important thing. You know a good lens captures more of the diffracted light than the poor ones. And that's true for electron density maps. The more data you have, the better data you have, the better the structure's going, the electron density map's going to look, the better the structure's going to look. All right, this is the Fourier synthesis, this equation. Now, of course you don't have to memorize it, but I'll tell you what this thing is telling you. All right, the electron density at a position x, y, z, in this electron density map, say at that position, right there, is the sum of all the diffraction amplitudes that you collected. That's the stuff you can actually collect. And associated with each one of those diffraction spots is a phase, And that's the problem. That's the so-called phase issue. So when you go to calculate an electron density map, that electron density is that position, x, y, z, is the sum over all the HKLs, the amplitudes, and the phase. But then you go to calculate the next position, and you use this Fourier synthesis. This is the lens. These are the data we collect, and it's the phase issue that we have to get. So associated with each one of those diffraction maxima is a phase. So the FHKL, this right here is called the structure factor amplitudes. We get that from our X-ray data. When we collect X-ray data, we are getting intensities of the scattered X-rays, which then you can calculate what the amplitudes are. This is the phase, and that's the problem. That's lost. And that's why we have to get around the phasing problem. Now, if you look back in the 50s and 60s, all this was being worked out. It's amazing how they were able to work it out. And what they used to do is multiple isomorphous replacement or single isomorphous replacement. Now what does that mean? Well, you take your crystal and you collect X-ray data. And then you move your crystal to some heavy atom like methylmercury chloride. Now, it doesn't always work this way. But if you're lucky, that protein will bind in the crystalline lattice that methylmercury, and then you collect data off that. And then you look at the two diffraction patterns, and the difference you'll see in the diffraction pattern arises from the heavy atom that's been bound in the crystalline lattice. And then there are these techniques called Patterson maps that you can figure out where are those heavy atoms bound in the crystalline lattice, and that gives you a handle on the phases. So multiple isomorphous replacement, single isomorphous replacement. We haven't done it in years, but it used to be something that we did all the time. But now there's this technique called difference Fourier maps, which I'm going to show you an example of. There's molecular replacement, which I will show you an example of. And then there's this mad phasing. Now the mad phasing is multiple wavelength anomalous dispersion. And in reality, it's very much similar to heavy atom derivatives, multiple isomorphous replacement and single isomorphous replacement. But instead of looking for heavy atom derivatives, you do something like make selenomethionine and put that into your protein. And you make selenomethionine labeled protein. And then you go to the synchrotron. You have to do this at the synchrotron because it's multiple wavelength. In our laboratory, in our diffraction equipment, it's only one wavelength. And in order to do this, you have to go to the synchrotron. This is mad phasing. This is something you do when you have a structure or a protein and you don't know what its structure looks like at all. It's brand new, you have no idea. But molecular replacement, I'll tell you about that. That's when you have a protein crystal where you know there's a homologous protein that's been solved. For example, some cytochrome you get from some bacteria. But you know in terms of amino acid sequence, it's going to be similar to another one whose structure is already known. That's called molecular replacement. And difference Fourier maps is what I'm going to tell you about right now. Difference Fourier maps. Supposing you have a structure of an enzyme and you solved it. And then you take the crystals of your enzyme and you have the structure known. And then you take the crystals and you soak in a ligand. And you then collect that data. And the crystal with the ligand, the same space group, same unit cell, right? So what you can do is take the diffraction pattern of the protein model that has no ligand bound and the diffraction of the protein model that has the ligand bound, and you can look at the differences and you can calculate an electron density map, which is a difference Fourier map. So for example, here's the reference structure, right? And here, so you collect the data off your reference structure. Then you collect the data off your target structure. You compare the difference in the diffraction patterns, and what you'll see is just basically electron density, where the new target has this right here. Now let me show you that in real life. This is an example from a difference Fourier experiment that was done in the laboratory. So we solved the structure of this PLP containing enzyme, and we're going to talk about PLP. But the structure first solved didn't have PLP bound in the active site. So then we got PLP bound and we collected X-ray data. You calculate a difference Fourier map, and all you see is the electron density that is different between those two models, between those two protein crystals. That's difference Fourier, and the drug companies do this all the time. They'll get a structure solved in like the HIV protease, right? You put an inhibitor in there, you've got it, then you do these difference maps and you can see how the drug bound, the inhibitor bound. So difference Fourier experiment. If you hear that term, they already know a lot. They're looking at just a little part of the map, these so-called FO minus FC maps. Now we have just a few minutes to talk about molecular replacement. And this happens supposing you solve the structure of galactose mu rotase from a bacteria. And it crystallized in this space group with those unit cell dimensions. And then you're interested in looking at the human enzyme. And you know, in terms of amino acid sequence, they're pretty similar. But your human enzyme crystallized in a different space group and different unit cell dimensions. So you can use this technique called molecular replacement. Here, for example, is the target of your unknown. So you know this is the structure. You crystallize that target unknown, and you collect the data, right? And then you have a diffraction pattern. And then what you do is you take your reference molecule and you start putting it in the unit cell. So the reference would be, for example, the bacterial galactose mu rotase. And you position that model into the unit cell of your target structure, and then you compare the diffraction pattern that you had with the, in your model here versus what you actually collected. And that doesn't look too good together. So then you move the molecule in the unit cell and you calculate what the diffraction pattern would look like, and you compare it to the one you actually collected. Not so good. You keep going, and finally what happens is you get a good agreement between the diffraction pattern that you measured and the one that you're calculating. You can do this with the reference molecule. And then you'll get this map and you'll be going along, and for example, supposing there's a tryptophan in the reference molecule, but in your structure, the target, there's an arginine. If you did it right, you'll be coming along and you can see the electron density for the arginine. Molecular replacement. So if you hear that term, it means that the structure that was solved is already similar to something that's been solved and is in the protein data bank. All right, so we have about a minute to go, and I think we'll end here and talk about mad phasing, and then we're going to talk about what do you look for when you're looking at structures and deposits in the protein data bank. All right, see you next time.